Hello students, today we are learn about protein purification. The protein purification is a series of process to isolate desired protein from complex mixture, that is cell, tissue or whole organism. The purification process separates the protein and non-protein parts of the mixture and finally separates the desired protein from all other proteins. The protein separation depends on protein size, physicochemical properties, that is isoelectric pH, binding affinity and biological activity of that particular protein. The proteins can be analyzed and identified by mass spectroscopy. The purified protein can be used to determine amino acid sequence for some drug discoveries and some for industrial purposes. As we know, some proteins are intracellular and some proteins are extracellular. If the proteins is not secreted by the organism into surrounding solution, the first step of each purification is cell disruption of cells containing protein. Depending on the source, protein has to be brought into the solution by breaking the tissue or cell containing it. So before studying protein purification technique, we have to learn about cell disruption methods. The overall goal in cell disruption is to obtain the intracellular fluid without disturbing any of its other component. The different cells have different structure, hence they require different methods for cell disruption. The cell disruption methods are initially categorized in two different categories, that is mechanical method and non-mechanical method. Mechanical method includes blenders, grinding with abrasive, presses, sonication. And non-mechanical method includes enzymatic method and chemical method. First of all, we will see mechanical methods. Mechanical methods require some sort of force to separate out intracellular protein without addition of enzymes or any other chemicals. The first mechanical method is blenders. The blenders can be used to disturb the cell wall. This is the same process used by centrifugation, which separates materials suspended in liquid medium. The another mechanical method is grinding with abrasive. It is also possible to achieve cell disruption by grinding with mortar and pestle. This method is used with plant samples that have been frozen in liquid nitrogen. Once the cell wall has been disrupted, the solvents are added to extract the biological molecule. These two techniques are very traditional methods used for cell lysis, but some disadvantages occurs at the time of their use. One important disadvantage is sometimes protein get denatured due to more heating. So another methods for cell disruption is cell presses. For cell presses, French press is an apparatus used for the disruption of the plasma membrane of cells by passing them through narrow valve under high pressure. The French press consists of hydraulic pump that drives a piston. The piston forces the liquid sample through a tiny valve under high pressure. Whenever sample passes through the valve, the cellular disruption takes place by shear force. The another method is sonication. In this method, sound energy is used to agitate the particles in sample. The sonication is performed by using an ultrasonic bath or sonicator probe, hence it is also known as sonicator. The power source attached to the probe generates sound energy. By introducing high power ultrasound into the liquid medium, the sound waves are transmitted in the fluid and creates alternative high pressure and low pressure, that is compression and rarefaction. Due to this compression and rarefaction, the small bubbles are formed in the sample and this process is known as cavitation. Due to this cavitation, it induces shear stress on the cell wall of cells and hence cell get rupture. Then non-mechanical methods. Non-mechanical methods are further divided into three classes, that is physical method, chemical methods and enzymatic methods. First, physical method. that osmotic lysis of cells takes place. It is through the process of osmosis, water can be entered into the cell, which causing its volume to increase up to the point at which cell can burst. Now some chemical methods. We can use some organic solvent, for example benzene, toluene, xylene, octanol. DMSO also can be used to permeate the cell wall. Next, some detergents, for example SDS, that is sodium dodecyl sulfate, sodium sulfonate, CTAM, Triton X100. These detergents are capable of interacting both water and lipids. It solubilizes the lipids in the cell wall and forms micelles in the water. Next, enzymatic methods. Use digestive enzymes which will decompose the microbial cell wall. For example, lysozyme. It hydrolyzes beta 1,4 linkage of cell wall of gram-positive bacteria. But the cell wall of gram-negative bacteria differs from the cell wall of gram-positive bacteria. So, lysozyme is not very efficient in the case of gram-negative cell wall. EDTA can be used to gram-negative bacteria. It chelates the cation which leaves holes in the cell wall. In this way, EDTA can lyse the cell. Thank you.